Last lecture we talked about independent assortment, and in this lecture we're going to talk about what happens when we cross two true breeding individuals with one another, and then we take the offspring of that cross and we cross those as well. So here we have our true breeding yellow round genotype for this pea plant. Okay, so the capital Y means that it is yellow, and the capital R means that it is round. Okay. This particular organism, it's been crossed sort of to itself again and again and again. That's why it's referred to as true breeding. So what gametes can we make from this particular organism? Basically capital Y and capital R, it doesn't matter how alignment happens. The only thing you're going to be able to make from this is capital Y, capital R, because that's the only alleles that it has to contribute. Okay, you can't have a lowercase r here because it only has capital R's. Now let's look at a true breeding green and wrinkled. The lowercase y means that it's going to be green. The lowercase r means that it's going to be wrinkled. And what gametes can we make from this? Well, just lowercase y, lowercase r, because again, that's the only thing that it has to contribute. But what happens, what kind of organism is going to result when we cross these two, when we take a true breeding yellow round and breed it with a true breeding green wrinkled organism? What you're going to wind up with is a heterozygote. Okay, a capital Y, capital R, lowercase y, lowercase r. This is going to be heterozygous for both traits. Okay, and what kind of gametes can this heterozygous individual make? Well, let's go ahead and replicate our DNA here. And so now we have sort of two copies of each. And we're going to have them get together in prophase. Maybe crossing over happens. We're not going to worry about that for this case, though. And they're going to generate these four gametes, assuming that the alignment happened like this. And the gametes you get from this are capital Y, capital R, lowercase y, lowercase r. These are your four gametes. Two of those are the same, so we'll go ahead and not even worry about those. So I just kind of knocked out these middle ones because you don't need to count them twice. Okay, but what if instead of having blue on the left and red on the right, you had these top ones switch? So now you have blue on the right and red on the left. What if your chromosomes light up like this instead? Or what if your tetrad formed like this instead? So now which gametes can we make from this cross? Or from this meiotic division? Well, it's a little bit different now, right? Now we can have lowercase I, lowercase r, capital Y, lowercase r. And again, two of these are the same, so I just knocked those out. You put that together from the gametes that we had before, and you wind up with sort of these four possible combinations of Y's and R's. And by the way, you don't have to do this every single time. You can kind of just put together all of the combinations. So you have a capital Y and a lowercase y. How can those fit together with capital R and lowercase r? These are all four possible combinations that you can have of these. Again, you don't need to go through the whole process that we went through. I just want to make sure it's crystal clear, where did these letters come from? How does independent assortment have an impact on what we're doing? So now the next thing that we're going to do is we're going to cross one of these individuals back to itself. Actually, what Mendel did is he crossed many of these individuals back to themselves. So that first true breeding generation was called the P generation. And now we are here in what's referred to as the F1 generation. So he's taking two F1 individuals and crossing them back to each other. And what you do there is called a dihybrid cross. Okay, so here was our first generation, our two true breeding individuals, which produced our heterozygous individual, which, by the way, has all of the dominant characteristics. It has all the dominant phenotypes. But then we're going to cross it back to each other in this dihybrid cross. So in a dihybrid cross, you're taking all possible gametes that you can make and crossing them with all the other possible gametes that you can make. Okay, so here are all of our possible gametes. Here are all of our possible gametes on the left, all our possible gametes on the top. And like I said before, you just bring them down. So this one goes down into each row, and these ones go across into each column. And you see what individuals are being generated from this. Okay, so the phenotypes that Mendel saw again and again and again when he did this type of cross is he saw 9 dominant for one characteristic individual. Okay, so these are yellow and they are round. Then he saw 3 that were dominant for one characteristic but recessive for the other. These are green, which is the recessive trait, but they're also they're round and not wrinkled, which is the dominant trait. He also saw 3 of the other way, where this is yellow, which is the dominant trait, but wrinkled, which is the recessive trait. And he saw 1 which was completely recessive. Okay, it is green, which is recessive. It is wrinkled, which is recessive. And if you look at our Punnett square from our dihybrid cross here, these numbers should make sense to you. You can actually just count them up. So let's say we're looking for the dominant one. I just count 1, 2, 3, 4, 5, 6, 7, 8. I saw 9 of these. And let's look for these 3. I see 1, 2, 3 of these. Let's look for these 3. I count 1, 2, 3 of those. And this last completely recessive one is down here. So by the way, Mendel didn't see this exact pattern, but he saw sort of large multiples of this exact pattern again and again and again. You had 9 times more of the dominant phenotype than you had of the completely recessive phenotype. Okay, and Mendel didn't know about Punnett squares, I don't believe. He didn't have access to sort of this formulation of things, but he was able to figure a lot of this stuff out by just actually doing the experiments. So let's do one more example. Run through an abstraction of this idea really quick. There is no allele for text being a particular color. It was just easier to show you guys this way. So if you have a capital A anywhere, the text is going to be black. That is the dominant allele. If you have a capital T anywhere, the text is going to be highlight blue. That is the dominant allele. Lowercase a is green text, and a lowercase t, there is no highlight. So we're crossing 2 individuals who are heterozygous for each trait. These are the possible alleles that each individual can make, and we just set these up along the 2 sides of our Punnett square. So if this sperm fertilizes this egg, what's that individual going to look like? If this sperm fertilizes this egg, what's that individual going to look like? And again, you just bring every single one of these down into every single row, and you bring these across into every single column. And when you combine all of these things, you see that same pattern that we saw with pea plants. And we can count them up. So let's count all the ones that have black text that are highlight blue. I count 1, 2, 3, 4, 5, 6, 7, 8, 9 of those. So 9 are expressing our dominant trait. Now let's count all of the ones that are black text and not highlight. 1, 2, 3. 3 have 1 recessive trait and 1 dominant trait. Let's count all that are highlight but with green text. I count 1, 2, 3. And let's count all that are double recessive or green text with no highlight. 1. So when you do these dihybrid crosses, you wind up with a 9 to 3 to 3 to 1 ratio. I'm not going to ask you about that number specifically, probably, but I'm almost certainly going to ask you to do a dihybrid cross like this one.